Good afternoon, ladies and gentlemen. Welcome to this session, which is on sepsis, the immune response. We have great speeches lined up for the afternoon. My name is Kodo Said. I'm a microbiologist from Southampton, UK, and my colleague Lenny Dede from Utrecht. We would like to welcome our first speaker, Prof. Merv Singh from UCL.
Right, thank you, Kodo. Hi, let's see if this works. Aha. Oh, let's go back one. Hang on a minute. Right, thank you very much indeed. Right, I declare right at the outset I am not an immunologist. I'm a very sort of lay immunologist. I like mitochondria, but the two are actually quite interesting together. Right, okay, we know, and you'll hear from our eminent speakers who follow me, that sepsis is associated with both immune activation, but importantly, immune suppression. At the same time, there's a nice association of bioenergetic dysfunction, mitochondrial dysfunction in multiple organs. Are the two related? I think the answer is probably. Immune cells are complicated because, they're circulating around in the blood and importantly in the tissues, and we don't really look at what's happening in the tissues, but in the blood there's a whole variety, a whole load of different types of cell, and within T cells, for example, there are the different subsets. So we've got lots of things going on within the bloodstream. And it gets even more complicated. You can break things down even further. And as I'll demonstrate, I'll just show a few bits of data later on, but these subsets differ both in number and activity during the septic process. I know Manu, a lot of his work has been related to that. The good news about mitochondria, they're ubiquitous, they're everywhere except in red blood cells, and as you can see here, it's not just about providing ATP, there's a whole load of other functions, and we know in sepsis, many of these things become dysregulated, abnormal, whatever, and so therefore there's a nice association with mitochondrial dysfunction. It doesn't prove cause and effect though. And in recent years, there's been lots of excitement in the immunology world about the role of mitochondria, the powerhouses, a central hub, etc., etc. So the question is, how do they affect immune function? And mitochondria regulate the activation, the differentiation, and the survival of immune cells. They release things. I'll go into a bit more detail in the next few minutes, but things like mitochondrial DNA, mitochondrial reactive oxygen species that regulate immune cell transcription, and a whole variety of mitochondrial activities also modulate immune function. So, depending on the type of substrate being used, be it, for example, glucose or fatty acids, dynamics, mitophagy, etc., can influence immune functionality. Mechanisms, I'm sorry, it's my Mac formatting's just been distorted by the computer system here. Hey, that's life. So as an example, you get alterations in metabolic pathways, the Krebs cycle, the tricarboxylic acid cycle, oxidative phosphorylation, fatty acid oxidation. And so, for example, the pro-inflammatory M1 macrophage, the Krebs cycle doesn't work that well. On the other hand, the anti-inflammatory M2 macrophage goes much more towards fat utilization, beta oxidation of fat. As I'll show you, the mitochondria can activate different components. For example, the inflammasome, fission and fusion seems to be important, and there seems to be also a component of signaling. So the mitochondria and the endoplasmic reticulum live very close to one another anatomically, and there seems to be some sort of interaction signaling that can affect immune function. As I mentioned, the monocyte can differentiate into pro or anti-inflammatory. The pro-inflammatory, there's much more in the way of glycolysis, mitochondrial ROS production, and a decrease in oxidative phosphorylation. On the other hand, anti-inflammatory, I mentioned beta oxidation of fat, but oxidative phosphorylation increases, AMP kinase also increases. It gets even more complex when you look at the T cell. So a resting, naive T cell uses at least 50% of its energy substrate. The ATP comes from oxidative phosphorylation. When it's activated, there's a big surge in glycolytic capacity. So to meet the acute need, inflammation, glycolysis upregulates to generate more ATP. However, depending on which T cell subset, the T helper cells continue using more glycolysis, whereas Tregs, memory T cells, again switch more back towards using oxidative phosphorylation and fatty acid oxidation. So you can see there are differences even within the same lineage. So mitochondria, essentially, when they're damaged, if there's tissue damage, they can be released, and the mitochondria can release components which, the mitochondria themselves activate TLR9. You also get other things like formal peptide, for example, which can stimulate receptors, TFAM, etc., and that can upregulate pro-inflammatory cytokine genes, so you get increased transcriptions, so you get an increase in chemotaxis, cytokine production, and so forth. So the mitochondria can be a direct stimulus outside the immune cell and acting on it. Intact mitochondria, too, from damaged cells, can release ATP, and ATP stimulates the P2X7 receptor, which in turn activates the inflammasome, and so interleukin-1, interleukin-18 can also be activated by mitochondria. Endotoxin can also, through mitochondria, have effects on inflammation. So you get an increase or dysfunction of the oxidative phosphorylation, the electron transport chain, and that causes a surge in mitochondrial ROS production. I won't go, because of time, through the mechanisms, but the net result of that is also activation of the inflammasome, release of interleukin-1 and 18. There's this nice article which looked at neutrophils, because most of the work in the literature has been focused on monocytes, T cells, etc. Not that much on neutrophils, but there was this nice review, and you can see the different aspects of neutrophil functionality, be it differentiation, phagocytosis, net formation, uses different metabolic pathways. So there are relatively few mitochondria in neutrophils. However, they do seem to play an important role in some of the functions of the neutrophil. With sepsis, you get, obviously, a rise in neutrophils most of the time. Again, what we're measuring is what's circulating rather than the neutrophils that are marginated into the pneumonic lung or the peritoneum or whatever. But in those circulating neutrophils, there's impaired migration, impaired phagocytosis, delayed apoptosis. There have been studies looking at ex vivo stimulating neutrophils with LPS and causing a dysfunction in chemotaxis, and that's associated with an inhibition of the electron transport chain. This is a very old trial. This is from the GLUE grant, and basically they took healthy volunteers, they gave them endotoxin, took blood samples over the next 24 hours, and then they looked at transcription of what was available at the time, and I can't remember, 1,500 genes I think they were measured. But interestingly, in the paper, what they highlighted were genes related to the mitochondria, so complexes 1, 3, and 5 of the electron transport chain, pyruvate dehydrogenase, and the permeability transition pore, which opens up and then can trigger apoptotic sort of cell death pathways. You can see blue downregulated. So the genes associated with these different complexes, proteins, etc., were downregulated, blue, 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 blue. So even in these healthy volunteers, again, paradoxically, you think, oh, they'll be fighting, they need more energy, but the signals being sent out was actually to suppress. This is, I helped Didier Payen and his group years ago, and it was a nice study looking at peripheral blood monocytes. And basically, if you took from healthy volunteers, incubated them with plasma from healthy volunteers or plasma from septic patients, you could see the oxygen consumption overall was significantly reduced, but the non-coupled respiration, so in other words, not going towards ATP production, was significantly greater. So even though there was an overall reduction in ATP production, as a percentage of total oxygen consumption, it was significantly reduced. There was also this, I think, interesting figure. This is in healthy volunteers, and you can take them at rest and then stimulate them with ADP, and you see an increase in oxygen consumption. However, when you did the same but incubated in septic plasma at day 1 of ICU admission, day 7 and day 28, there's a different picture. So at day 1, there's an increase in the baseline, but with stimulation, you can see it's quite markedly suppressed. But over time, as the patients are getting better, over day 7 and day 28, you can see that there's a progressive return to what the control cells looked like. Not quite back to normal, but going in that direction. Some nice and interesting paper from a group in Brazil where they looked at the proteome of the septic monocyte, and blue is downregulated, red is upregulated, and again, this association. You've got cytokine signaling, interferon signaling, MHC class 2, HLA-DR, etc., downregulated. At the same time, a downregulation of the Krebs cycle, mitochondrial electron transport chain, fatty acid oxidation, yet at the same time, glycolysis was upregulated. And they showed this variation over time. They took sequential samples through the patient's illness, and you can see glycolysis high initially in the septic patients and then fell during the recovery phase, and the opposite was seen. There was an initial mitochondrial depression which recovered and normalized during the recovery phase. That's actually a very nice paper from 2 of the speakers in this session. So we've got Peter and Tom in the front row, and I won't go through the paper in any detail, but here's the conclusion. And they were looking at this concept of immunoparalysis and is there a relationship with energy metabolism. And they found, as I described earlier, overall there was this shift from OXPHOS to glycolysis. Leukocytes with severe sepsis had these metabolic bioenergetic defects, and these cells couldn't respond as well ex vivo to stimulation challenges. So analogous to this immunometabolic paralysis. Thirdly, therapeutic approaches that reverse this defect might represent a novel therapeutic approach, and so they found a partial response with interferon gamma. I'll show one picture from their work. It was, again, ex vivo oxygen consumption in peripheral blood monocytes from healthy controls and septic patients, and you can see there's a clear difference, especially when maximally stimulated, between samples from healthy volunteers and from septic patients. So in summary, yes, mitochondria are involved in the functionality of all immune cell types, though it does vary from type to type, and clearly there's a difference when the cell is activated to when they're quiescent. There is an association, an interesting association, with immune suppression in sepsis, though we've yet to see the definitive study where you give something to activate the mitochondria, which then restores immune functionality. On that note, I'll finish. Thank you very much indeed for listening.
Thanks, Mev, for the interesting talk. And on time, we have time for a couple of questions. Anyone on the floor? Mev, I'm interested. You mentioned very little is studied in neutrophil, mostly in monocyte macrophages. What about platelets?
There have been a few studies, well, again, in looking at platelet dysfunction and mitochondrial activity, and to be fair, there are conflicting results. So, again, is it methodological? Because, again, it's the challenge, and, I think hopefully the immunologists and the mitochondriacs will acknowledge that when you take something out of the normal milieu and do things ex vivo, how representative is it of what's happening in vivo? So we're making the assumptions. And so I think a lot of variations you see in the literature, I would argue probably, obviously, different patients, et cetera, but I think there are methodological challenges too. So I think I'll sit on the fence on platelets.
Thank you.
Thanks very much, Murray.
Lovely.
Really great. The next speaker also needs very little introduction and is the only speaker we'll see twice in this session. So I think, Tom, that may make you the expertest expert of them all. Tom's going to tell us about age-dependent host response dysregulation. Tom, the floor is yours.
Thank you very much, Lenny. Let me see whether I can get the slides on. Oh, okay. Now do I get back? Yeah. Okay. Yep. Okay. Welcome, everybody. My talk's about age-dependent host immune aberrations in sepsis. And let me first start by showing this graph from Germany, but I could show this from any country that has a relatively high income. That is, that mortality or the incidence of sepsis, in particular in the older ages, as you can see on this slide, and this relates to both women and men. And if you look at mortality of sepsis, this is also age-dependent. And in the mid-decades, let's say between 30 and 50 or so, this is the more comorbidities you have, the more likely you are to die from sepsis. But eventually, if you reach a very old age, it doesn't really matter anymore. Your likelihood of dying increases proportionally with your age. And this has to do with a gradual decline of your immune system. And this is illustrated on this graph by Dr. Monneray, explaining the concept a little bit of immunosenescence, which is this gradual deterioration of your immune function. So when you are young, on the left-hand side of this slide, you see this dotted line, which is sort of the immune homeostasis line. And you're very easy to go back to immune homeostasis when you're young, even if you have a septic event. But when you are older, you're actually already beyond this line of immune homeostasis. And then you're more vulnerable. And once you are hit with a septic insult, you are less likely to return to a normal situation. So this is basically a double hit. And if you look at immunosenescence, this relates to both the innate immune system and the adaptive immune system. So if you look at this upper part here, so this is the innate immune system, and there are several functions that are decreased there, and in particular neutrophil functions, like formation of neutrophil extracellular traps, which are important for antimicrobial defense, and also oxidative burst. Phagocytosis is declined. But remarkably, with respect to the innate immune system, there's also a state when you get older that has been referred to as inflammaging, which relates to the fact that people that are old seem to have a chronic pro-inflammatory state, which you can detect, for example, by elevated CRP, C-reactive protein levels, and also some pro-inflammatory cytokines. And if you look at the adaptive immune system, that's below here, that's also declined, the function thereof, in patients who are aging, so at higher age. So there's not enough time to discuss all of these phenomena, but make sure one of the take-home messages is that your immune function is declining, and that starts beyond the age of 60 approximately, and it heavily takes on beyond 65 and 70. Now, this is a study our group did a few years ago with patients that are admitted to a general ward with community-acquired pneumonia. And we split here the patients into those below 50 years of age, so the young group, and above 80 years of age, the old group. And sure enough, the older patients had reduced CRP levels, whereas we know from healthy elderly people that they have elevated CRP levels. But if you then look at other inflammatory response, and in particular at the cytokine responses, there is not much differences between the two age groups. So even the very young patients, they have similar pro-inflammatory cytokine profiles as compared to the older patients, and this also relates to the anti-inflammatory cytokines like interleukin-10 and IL-1RA, interleukin-1 receptor antagonist. And this is sort of confirmed in different studies, and I took one of them. So this is from the GenEMS cohort. So this cohort involves patients that are a little bit more sick. So they are presented to the ER, and part of them also have sepsis with community-acquired pneumonia. And you see here the cytokines, TNF, IL-6, IL-10, and basically there's no difference between different age decades, as you can see on this slide. So the systemic pro-inflammatory cytokine response does not seem to differ that much between age groups. This is another set of data, and we're switching now to the more severely ill group. So these are patients with sepsis, and this was studied in different age groups, as you can see on the right-hand side of the slide. And they looked at different inflammatory proteins, and I copied this graph showing interleukin-1 receptor antagonist, NGAL, which is a neutrophil degranulation product, TNF receptor 1, the soluble version thereof, and CRP. And you can see then that NGAL and the soluble TNF receptor 1 are higher in the elderly patients, and the other proteins, the other mediators, are also somewhat elevated in the older patients, but it's not so striking. So the differences are not that tremendous as you might have expected from the fact that these immune systems are way different from each other when you compare young people with old people. Now I come to a study that we did in Amsterdam and Utrecht. This is part of the MARS cohort, which was a cohort which enrolled 8,000 patients in total between 2011 and 2015 in the two ICUs of Amsterdam UMC and Utrecht UMC. So it was a collaborative project. And what Erik Michels, one of our PhD students, did was stratify the patients according to decades, ages, and you see the numbers, quite high patient numbers. They are listed here. And what you can see from this slide is that, first of all, I just copied the highlights. So not unexpectedly, this is the Charleston Comorbidity Index. So patients that are older have more comorbidities. Well, duh, that's what you would expect. And then you see the severity of illness on presentation. Well, the Apache score doesn't differ, but that contains a lot of chronic comorbidity items. But if you look at the SOFA score, which is more related to the acuteness of the illness and the percentage of shock, the elderly patients are more sick and they more often have shock. Not surprisingly, there was an increase in sepsis-associated mortality in the older decades. So you see the blue line, which contains the patients that are above 70, and there's clearly an age-dependent increase in mortality in the ICU with regard to sepsis. And now comes the topic of this talk is the immune response. And what Erik Michels did, he looked at quite a few protein host response markers, and he also looked at gene expression profiles, and I'm going to allude to that in the final part of my talk. So these are systemic inflammatory response markers, CRP, you're all well familiar of, MMP8, matrix metalloproteinase 8, and the inhibitor thereof, TIMP1. And what you can appreciate, what he did is he looked at 3 different p-values. So he looked at the unadjusted p-value, seeing whether there's age-related differences in the host response. Then he looked at severity-adjusted p-value. So I alluded to the fact that the older people were more severely ill, and biomarker levels tend to increase or become more disturbed proportionally to the severity of illness. So we did a model that adjusted for that. And finally, we also adjusted fully, which means that we basically adjusted for all baseline differences between the different age groups, except for the age, just to see what age does. Maybe not so physiologically meaningful, but the second adjusted p-value is. Well, CRP is higher in the elderly patients, but the other products, there's not much difference between the age groups. And the same is true for cytokines. So this is IL-6, IL-8, and IL-10. And this is sort of corresponding with what I showed for the pneumonia. So these were not critically ill pneumonia patients in the previous slides. These are critically ill patients admitted to the ICU with sepsis. But also there, the cytokine response does not differ between ages, in spite of the fact that these elderly patients are more severely ill and are more often dying. And this is relatively new because not many people have looked at this. So this is the endothelial cell response. And you might appreciate that endothelial cell activation and disturbance of the function of endothelial cells is a pretty important phenomenon in the pathogenesis of sepsis. And what we did here is we looked at endothelial cell activation markers, E-selectin, ICAM1, and fractalkine. And what you can see in this slide is that the blue boxes, so the older patients, are lower than the red boxes, the younger patients. So older patients have a less profound endothelial cell activation in sepsis, in spite of the fact that they're more severely ill. And then look at endothelial barrier function. So you can do this in particularly by looking at the ratio between angiopoietin 1 and angiopoietin 2. The dotted line in the graphs are the normal values, so the reference values. So the further you are away from the dotted line, the more disturbed you are. And again, here you see that actually the elderly patients do a little bit better. So their barrier function is not as disturbed as in younger patients. And then with regards to coagulation, there was not a lot of differences, except for platelets. So remarkably, platelets were a little bit on the higher end in older patients, but that's just a phenomenon we observed. So usually when you have DIC or so, your platelet counts are very low, and low platelet counts are actually associated with worse outcome. But these are all platelet counts that are not very disturbed, even in the young patient group with the lowest platelet counts. This is still, the median is almost normal, so it's not like that this is very important data. But when you look at the regular readouts for coagulation activation, so prothrombin time, D-dimer, and the anticoagulant proteins, antithrombin and protein C, this is all similar. So what you would expect in patients with sepsis actually depicted on this slide. You have a prolonged prothrombin time, elevated D-dimers, and reduced anticoagulant protein levels, antithrombin protein C. But this is all similar between age groups. And then finally, we looked at blood leukocyte transcriptomes. And then we made two groups, a young group below 50 and an old group above 70. This is our own cohort, so this is the Mars cohort, and this is a validation cohort coming from the UK, so this is a community-acquired pneumonia cohort, severely ill, from Julian Knight's group. You may know the publication. And what we see here is the blue bars are lower in old patients, and the red bars are higher in old patients, and what we did is we did reactome pathway analysis. So you group the genes that are differentially expressed, and you group them into functional pathways. And this is what you show, what you see here on this slide. And what's the take-home message here is that genes that are associated with innate immune signaling, that's here, and also with adaptive immunity, are lower in those patients that are older when compared to patients that are younger. But there's the genes in blood leukocytes that associate with endothelial cell activation, in spite of the fact that the markers in plasma of endothelial cell activation were actually lower in patients that are older. The leukocytes show signs of enhanced interaction with endothelial cells. And also, genes relating to coagulation activation in leukocytes were enhanced more in patients that are older. Now, we asked ourselves the question, what is now already present prior to developing sepsis, and what is sepsis driven in these differentially expressed genes? And this is a cohort we found in public space. So these are healthy people separated between below 50 and above 70. And then you can see that some of the changes that we see are sepsis driven, sepsis specific, and in particular relates to the innate immune system, because in accordance with the term inflammation, healthy older people have enhanced expression of genes that relate to innate immune signaling. But adaptive immune function is already decreased when it comes down to blood leukocyte transcriptomes. So the reduction in adaptive immunity is not sepsis specific, but it's maybe an aggravation of what's already present. And then the other phenomena are partly present already in healthy people. So I come to my final slide, a summary slide. What I've showed you, and this does not come as a surprise, sepsis incidence and mortality increases with age. What I've showed you in patients that are not on the ICU but admitted to a general ward with pneumonia, but also patients with sepsis admitted to the ICU, the systemic cytokine response and in general the systemic inflammatory response does not differ too much across different ages, although the older patients are sicker at presentation. And I've showed you that the older patients have less profound endothelial cell activation and less profound endothelial barrier function disturbance. And then finally, the blood transcriptomes do show differences that are age-specific, but some of these changes are already pre-existent, and in particular those that relate to adaptive immunity, that's reduced. And some of these changes are sepsis-specific or induced-driven, and these relate to innate immunity. Thank you very much for your attention.
Thanks very much, Tom, for that presentation. Lots to unpick. Are there questions from the audience? Yes, in the middle there, the gentleman. I hope there's a microphone. All right, in the front here. There's another hand there in the back. Yeah, okay. Here in the front first.
Thank you. That was really interesting. I was curious if you've stratified your results by metabolic comorbidities, diabetes versus non-diabetic patients, given the effects of hyperglycemia on immune system function.
Yeah, we've done that a few years ago. There's actually 2 papers about this. And diabetes per se does not influence the host response to a significant extent once you are in the ICU with sepsis. Obviously, diabetes makes you more vulnerable to develop sepsis, but once you are in the ICU with diabetes, it doesn't make a difference anymore.
And there was one gentleman in the back with a question, yes?
Did you look at the role of vitamin D in these patients? Because we know that the elder people have a low vitamin D, and that could lower your immune response if it's normal, but increased enormously when it's low.
We have not looked at vitamin D, I'm sorry, so it could still be measured if you want to, but we haven't done that. Thank you.
Well, I would like that.
I can see opportunities for collaboration coming here. We're going to withhold further questions until the discussion in the middle of this session. So if you have any questions, don't let them slip out of your head. Remind them, because after the next 2 talks, we'll have some time for extra questions. So don't hesitate to ask your questions then.
Yeah, the next speaker is Peter Pickers from Nijmegen in the Netherlands, about gender and immune response.
Thank you very much, Mr. Chairman. So following the talk on the influence of age, I will now talk about the influence of gender. And if I talk about gender, of course, I have to focus on sex hormones and about men and women being different. So I apologize up front for all the political incorrect stuff that may come. I do mean well. So to give you a short history on how men and women were different and how we're actually growing towards each other. For the women, actually, it was 1830 when women were first allowed to wear trousers in public, and the 1919s or 1920 when women could vote for the first time in general elections. And then a couple of years later, there was a huge increase in lung cancer in women because they also started to smoke, as the men only did before that. And from the other side, it's amazing to know that the first man that underwent a sex change operation was operated in 1931. And then in the 60s, of course, the Beatles and the Rolling Stones made long hair popular among men. And in the 1990s, we had the invention of the word metrosexual. And this is a heterosexual man with feelings for beauty and style, and he uses cosmetics, and he spends a lot of money on clothes. So we are coming together. There's almost no difference anymore between men and women. But there are, of course, we know that there are huge differences between men and women. And this is just an illustration of neurological differences. But also, I will tell you indeed that there are differences in the immune response. So I will focus on sepsis. And the intriguing part is that the treatment for sepsis is the same in both men and women. And is this correct, or should it be like this? And as a man, I have to admit that women do better in trauma, in cardiac infarction, in sepsis. And so you wonder, is the insult different in women? Is their response different, or are they just smarter? And so women do have a better prognosis as well. And this starts at the very, very beginning. In newborn babies, if you are a male baby, you're more than twice as likely to suffer from an infection compared to a female baby. And then over the whole lifespan, the incidence of sepsis is lower in women. And the mortality, if they do get sepsis, then their mortality is also lower if you compare that to men. And so these women are surviving, and then they write books. You can buy this. I looked it up at Amazon. It's $16, I think, My Survival After Sepsis. And the intriguing part is I could not find a book written by a man that survived sepsis. So this illustrates the huge differences. What could be the mechanism? Now, of course, there are these sex hormones. And indeed, of the female sex hormones, many beneficial effects have been shown. In animal studies, indeed, if you have like a global ischemic insult or systemic inflammation or focal injury in vital organs, indeed, if you give female hormones, you can mitigate this, and there are positive effects. So maybe these female hormones could be a therapeutic option in sepsis as well. Some people really believe in this stuff already. So this is a review that is extremely intriguing in my view. And they summarize that indeed the rationale to give female hormones to sepsis patients or all critically ill patients is very compelling. There's an extensive body of extremely supportive data. It's very persuasive, and this should be done routinely to all patients in the ICU that have critical illness. One little thing is, there are no clinical trials yet. There is observational data, and it seems like that if a man is severely ill, the tactic is pretend to be a woman. It's true, it's not a joke. So we measured female and male hormones in men with septic shock. And we found that indeed, this is first testosterone. And as you can see in the cream bar, this is the normal reference range, that most men are severely suppressed. And that it does come back at day 7 after septic shock. But so testosterone is much, much lower, while the female hormones, estradiol in this case, is elevated in almost all men and also tends to normalize again at day 7. So indeed, the response to critical illness is lower testosterone, increase the female hormones, like pretend to be a woman. And we know this for a long time already. This is indeed, again, a similar study, again, septic shock patients. And what was found indeed was that in patients, especially the non-survivors, the female hormones are higher. So this could be adaptive, and so it's a good response, but still it's not high enough to survive, so it's higher in non-survivor. And the male hormones are suppressed. And there are different ways how hormones and how these hormones are influenced and how the enzymes that are relevant are influenced by inflammation, for example. But it's good to know that almost all men with sepsis have suppression of their male hormone levels. So this is a huge effect. And also in COVID, this has been shown that if you have lower levels of testosterone, you're more likely to have a severe disease course and a higher mortality. And this is also associated with the inflammation during the observation. So the big question is this adaptive or is this maladaptive? If it's adaptive and the female hormones are good, and so there's an increase during sickness trying to save your body, then it might be a reason to give more female hormones to patients because the endogenous increase might not be enough and some people still die. If it's maladaptive, and for example, that the suppression of the testosterone levels is not a good response, then you might want to actually give male hormone because this depletion needs to be normalized. So the problem with all these observational studies is that these are all associations. And just to illustrate what that means is this is the association between the number of people who drowned by falling into a pool and the number of films Nicolas Cage appeared in. And there's a very, very good association. And I'm not sure if maybe the people that are a fan of Nicolas Cage, and it's unclear why this is, and people say, well, it has nothing to do with each other, but actually I found out that you can buy a blanket with the face of Nicolas Cage, and this might also explain this association. But we do not know what is cause and what is effect. And it's the same thing. So we have a lot of preclinical data showing the effects of these female and male hormones on all kinds of processes, inflammation, ischemia, perfusion effects, but we do not know actually what the effects are. There's one study, or a couple of studies analyzed here in patients with heart failure, that indeed if you give testosterone to these patients, that indeed you might benefit them a little bit, and so their 6-minute walking test was just a couple of meters more pronounced. But for example, we know, of course, in critically ill patients in the ICU that this anabolic steroid also aimed to increase muscle strength, etc., was meant to give people the possibility to come off the ventilator and leave the ICU, but what they found in contrast was that patients treated with this anabolic was that they were longer on mechanical ventilation and they stayed in the ICU longer, so it's not that easy. There is no sepsis trial using female hormones. This is actually a limit. This should be done. This is an interesting stuff. There's one trial that is ongoing at this moment in the United States that is giving female sex hormone to prevent bleeding and shock from bleeding. So this was actually sponsored. It's a 10 million grant from the Department of Defense in the USA, and it's of course interesting in the military to give a shot indeed to prevent dying from bleeding. So just imagine you are this macho soldier at the battlefield and then you're shot and the first thing that happens is that you get an injection of female hormones, but this might be beneficial. This whole grant is actually based mainly on one paper in rats, and indeed if you remove a large proportion of the circulating blood volume in these rats and you give them female hormones, the 6-hour survival improves tremendously. So there's different effects that might be beneficial. As you might know, we perform experimental human endotoxemia in our institute, and so we have healthy volunteers that come in and they look happy and on the left, and then we give them a little bit of E. coli endotoxin, and then they get sick for a couple of hours, and they have a huge increase in cytokine levels. And so this is a very well-controlled, reproducible model to study innate immunity. And so we have just completed a very large project. It's called the 100 LPS project, and we ended up with 113 volunteers, 50-50 females and males. And look at the differences. The female volunteers in this very controlled setting have a more pronounced pro-inflammatory response. TNF, IL-6 are higher, while the anti-inflammatory IL-10 is very, very similar. So the pro-inflammation is much more pronounced in females. And we gave endotoxin twice, then you have a tolerance effect. Well, the same you see at the second administration, and the amount of tolerance that develops is the same between men and women. Now, when we actually asked the women, are you using any systemic hormonal contraception? And then we divide it up. Indeed, it's the women that are on contraception that have the most pronounced pro-inflammatory response. And the women that are not using hormonal contraception, they look a little bit like the men already, and the men are the lowest ones, actually. So this remains true, and it seems that if you give more female hormones in the form of contraception, you can actually boost the immune system. And so we also measured the circulating endogenous female and male hormones. And as you can see on the right in the graphs, in red, are the females that are on contraceptives. And that means that if you are receiving more hormones by this pill, your own endogenous levels go down. And so we found indeed negative associations with the pro-inflammatory responses, while in the men there were clearly positive associations with pro-inflammatory response. And so this suggests indeed that these hormones indeed directly affect the immune system. And so we took PBMCs from both male and female donors, and we exposed them to different levels of female and male hormones, and then exposed them to endotoxin. And as you can see, if you expose them to increasing concentrations of female hormones, their immune response becomes more pronounced, while testosterone does not have this effect. So it seems to be indeed that female hormones do improve your immunity right away. So my conclusions are there are many, many observational studies, and these are all associations. It's very unclear if it's adaptive or maladaptive. So we do not know at this moment that we should give the one or the other. There's no clear cause-effect link. We have no data on timing, dosing, etc., etc. And so a lot needs to be done. This is actually a tweet from our highly esteemed chair of the whole session, of the whole meeting here, that during the COVID pandemic, he tweeted, "I started a cocktail of anti-androgens and estrogens, and my prostate is fine, thanks, and the evidence is getting compelling." So indeed, there's also a lot of data on COVID course and outcomes related to these hormones. And I thought I just want to share with all you that his prostate is fine. So the future is bright. Thank you very much for your attention, and happy to take your questions.
Thank you very much, Peter. Any questions from anyone, please?
Please.
Yeah, can you please
Do you
introduce
have
yourself?
any studies on pregnant women?
No, I don't know that there are any studies on pregnant women, not in sepsis. I do know that in COVID, pregnancy was a risk factor. And I think it's mainly related to the fact that, as in obesity, the pulmonary mechanics are influenced by the pregnancy. And so I do think during pregnancy is like a unique state as well, that the woman is tolerating this fetus that has a different genetics. And so there is like a tolerance that is occurring, while at the same time, for most infections, they are not more susceptible. So it's not like everybody that is pregnant is getting infections. So they are, at the one hand, there is tolerance, and on the other hand, they still have an effective immunity. But I'm not aware of any real observational data in pregnant bacterial sepsis patients. Thanks.
Thank you, Peter. Lovely talk. Although I didn't want to know about Jean-Louis' prostate. The menopause, you know, because again, there's a talk, I remember studies years ago where, well, observational again, where cardiac arrest and trauma, pre-menopausal women did better than males, but after the menopause, they lost the advantage, survival advantage. Any thoughts, comments?
No, so this is one of the questions that I hope that would not be asked. The thing is, in sepsis, menopause, so indeed the advantage is staying, is remaining. And of course, following menopause, the female hormones go down, but still there is an advantage. So it's not only that, apparently. And so in sepsis, it's maintaining even for older women compared to...
They're healthier than men.
They do less crazy stuff, yeah.
Speaking personally, yeah.
Thank you. Peter, I have one question. Again, like you, I apologize if I'm not politically correct. Are you aware of any studies when there had been gender reassignment through hormone therapy that risk factor goes down or up or vice versa?
Yeah, no, I'm not aware, and I'm pretty sure it's not there. in these woke times, it's time for that kind of stuff, I think. It would be of interest. I think I'm afraid for the observational trials, you need huge numbers to show anything at all. But it would be of interest indeed to see if there are differences that might be occurring. Yeah. Thanks.
Once more, thanks.
All right.
Oh, is there a question?
One more?
Yes, thank you. Thank you for your nice presentation. I will ask you about iron, because it can have an effect in female, which iron stock may be lower, and then therefore they do less oxidative stress.
Yeah, so that's an interesting one as well, because indeed, especially in menstruating women, there might be an iron deficiency, and iron is also related to the immunity. And the idea about that is that indeed it is an adaptive. So if we give endotoxin to the healthy volunteers, we have an acute lowering of the iron as well. And the idea is that this is an adaptive response, and that bacteria, they need the iron also to multiply, and so that you might be limiting the infection by that. We actually did an endotoxin trial in which we give iron, or we gave iron binding compound, and we found no effects on immunity in that trial. And I'm not sure, I'm not convinced of the adaptive response that is so important for the bacteria to have iron to actually multiply and to indeed cause an infection. Because in the bloodstream there are not that many bacteria all the time, so it's a local thing. So it's not there, I guess, but what we have done in translational work, we did not see a very strong relationship between iron levels, or giving iron, or binding iron, and the immune response.
Thanks, Peter, for that, and thanks to the room for the very interesting discussion. Thank you. So then we go back to Tom for the last presentation before the discussion time, host response and causative pathogens.
Thank you, Lenny. So you're well aware of the fact that sepsis is an infectious condition, and in approximately 2/3 of cases you can find a causative pathogen in sepsis. 1/3 the causative pathogen is never found. But if you look at the causative pathogens in sepsis, bacteria are most frequently isolated, and then followed by fungi. Where the proportion of fungal infection has been increasing over the past decades, presumably due to the fact there's more immunocompromised patients. So the proportion of gram-positive versus gram-negative is approximately the same. There has been a period where gram-negatives were prevailing over the gram-positives, but now, most recently, it's approximately equal. So if you look at gram-positive versus gram-negative bacteria, they are very different. On the left-hand side, you see gram-positive bacteria, which have a very thick peptidoglycan layer, and the pro-inflammatory component, a pro-inflammatory component is lipoteichoic acid, which sticks out of this peptidoglycan layer. Now, gram-negative bacteria also have peptidoglycan, but it's a very thin layer, and their pro-inflammatory properties are determined by lipopolysaccharides, which are part of the outer membrane of gram-negative bacteria. And basically all pathogenic gram-negative bacteria have lipopolysaccharides, and makes them very pro-inflammatory when you test them in vitro conditions. In a first attempt to see whether the host response differs between gram-positive and gram-negative pathogens, we again went to the Mars consortium, the Mars cohort, so collaboration between Amsterdam UMC and Utrecht UMC. And Lisa Pereversova looked at differences between patients that presented with community-acquired pneumonia on the ICU with a pathogen that was assigned to this community-acquired pneumonia. So many patients do not have a pathogen, and in particular in CAP, 50% or so of all patients, there's no pathogen identified. So these were the patients in whom a pathogen was identified as causative, and we split them out between gram-positive and gram-negative, and there was no mortality differences between the two groups. So this is all critically ill patients, right? Then Lisa looked at the host response, and we separated these out into different pathophysiological domains. So this is the pro-inflammatory response and the cytokine response. And in blue, you see the gram-negatives, and in brownish, you see the gram-positive caps. And there was no difference between those two groups, with the exception of matrix metalloproteinase 8, which was slightly lower in the gram-negative group. But overall, these responses were fairly similar. And also, when we looked at other pathophysiological domains, which is the procoagulant response and the endothelial cell activation and function disturbance, so these are all mechanisms that are considered to be very important in the pathogenesis of sepsis, there were no differences between gram-positive and gram-negative community-acquired pneumonia. The dotted line in these graphs is always, again, the normal reference value, so the farther you're out from the dotted line, the more disturbed you are with regards to host response. And then we went on, because the sample size was big enough for this, to look at individual pathogens. And then it emerges a little bit that, some pathogens induce more changes in host response with regards to plasma host response protein biomarkers than others. And what you can appreciate from this slide, so this is again the pro-inflammatory cytokine domain, that the light blue boxes, which is E. coli, stand out a little bit as the most pro-inflammatory or the most cytokine-inducing pathogen in severe community-acquired pneumonia, so community-acquired pneumonia on the ICU. And second best is probably Streptococcus pneumoniae, which is the red boxes is here. So these two pathogens stand out a little bit. Overall, there's now differences across pathogens, and this is mainly driven by Strep pneumo and E. coli. Now, with regards to the other pathophysiological domains, this is procoagulant response and endothelial cell activation. In the upper panel, you see procoagulant response, and there's basically no differences between pathogens. But in the endothelial cell activation and disturbance of function domain, again, there's differences across pathogens. And again, E. coli and Strep pneumo stand out as the pathogens that cause the most host response aberrations when it comes down to measuring these protein biomarkers reflecting endothelial cell function and activation. Then what we also did, we looked at whole blood leukocyte transcriptomes. In gray, this is a PCA plot. In gray, you see the healthy controls. And then you see in red gram-positive and in blue gram-negative cap. And sure enough, there's a humongous overlap between these two groups, gram-positive, gram-negative. And there's not much difference. And this also is illustrated by this graph. So you see the volcano plots, which depicts all of the differentially expressed genes on the left-hand side between gram-positive pathogens and healthy condition and gram-negative bacteria and healthy condition. In blue, it's down-regulated genes. In red, there's up-regulated genes. And sure enough, there's a humongous genomic transcriptomic response induced by sepsis. But there's not a lot of difference between gram-positive, gram-negative community-acquired pneumonia. So this is the overlapping response. And the unique genes, these could not be allocated to functional pathways. So the conclusion is that the leukocyte transcriptomic response is largely similar between the two causative pathogen groups. Now let's now switch a little bit to a slightly different population where you might expect that if a pathogen has an effect on the host response that you measure in the blood, it's most likely to be detectable here. And this is patients with positive blood cultures. Now this is a relatively old study, 11 years old now, looking at patients with bacteremia, either gram-negative or gram-positive. And what comes out of this is the C-reactive protein and interleukin-6, which are higher in patients with gram-negative bacteremia, which may relate to the fact that gram-negative pathogens express lipopolysaccharide. And this is probably one of the most potent pro-inflammatory molecules that we know from biology. This is yet another study published a couple of years ago, also positive blood cultures. and now stratified according to gram-negative, gram-positive, and also culture-negative. And there was also fungal infections now included. If you look at the cytokine response on the left-hand side, interleukin-17 and tumor necrosis factor, again it stands out that the gram-negative bacteremia patients have higher cytokine levels than the gram-positive bacteremia patients, and also higher than fungi. But if you look at P-selectin, which is a marker for endothelial cell activation, but also for platelet activation, you see that the difference between gram-negative, gram-positive is not existing. And now here, the culture-negative seem to have a slightly higher levels of this mediator. Now, we did again a quite extensive study in the Mars cohort. So these are critically ill patients on the ICU with positive blood cultures. And this study was done by Joe Butler and Hessel-Peter Singers. And they looked at all different pathogens that were cultured from these blood bottles. You see here the different pathogens in different color codes. And here you see, this is 10 markers in this slide relating to systemic inflammation and cytokines. So interleukin-1 receptor antagonist, there's a few cytokines in the upper panel here. And in the lower panel, you have procalcitonin, soluble RAGE, tenascin C, soluble TREM1, and soluble CD163, which are all markers of inflammation-damage. And you see that these markers of inflammation, damage, and cytokines differ across pathogens. And what you can also tell is that the green boxes probably are highest or most disturbed, and the green boxes relate to E. coli. So this sort of corresponds a little bit with the CAP cohort, where we didn't look only at blood culture positive cases, but causative pathogens in general could also be cultured from sputum in the CAP patients. This is only blood culture positive. The other pathogen that stands out a little bit are the orange boxes, which is another gram-negative pathogen, which is Klebsiella. Now let's look at the other pathophysiological domains. So this is endothelial cell activation and disturbance, and also coagulation activation. And now it becomes a little bit less clear, although the orange boxes, which is Klebsiella, still are at the higher end. So there's an overall difference between pathogens, that's for sure. But it's slightly different with regards when we compare with systemic inflammation and cytokines. But the Klebsiella is really disturbed. But now also with regards to endothelial cell activation, and we thought this was a little bit surprising, fungal sepsis is also associated with more profound endothelial cell activation when compared to bacterial sepsis. And then finally, Joe and Hessel did a quite extensive investigation with regards to blood leukocyte transcriptomes. And this is the layout of what they did. They did a discovery cohort and a validation cohort. This is all blood leukocytes. The discovery was done with RNA sequencing, and the validation was done with the U219 microarray from Affymetrix. And they compared each pathogen group with non-infectious ICU patients, and then they did pathway analysis using the Reactome database. I alluded to that in my previous talk. So what Reactome does, and there are several other programs that do this, you look at differentially expressed genes, but these can add up to thousands and thousands of genes, and it doesn't make a lot of meaning when you look at these individual genes. So what Reactome does, it groups the genes in certain pathways, functional domains, and this is what Reactome does. So these are the mother pathways, or the father pathways, but they call them the mother pathways. And below that, there are several other pathways. And what I'm going to show you is what these pathogens do. And then there's, relative to non-infected ICU controls, there will be down-regulated pathways, and there will be up-regulated pathways. And this is the number of genes that are in each pathway. And I'm only going to show you the genes that were significant, or the pathways that were significantly altered, if they were both in the discovery and in the validation cohort altered in the same direction to a significant extent. Now, this is a slide that alludes to the fact that, and this came a little bit as a surprise to us, because you have to realize that on the ICU there's so much background noise, so to speak. These people are very, very sick, undergo all kinds of interventions, but nonetheless the pathogen explained a fair amount of the variation in the blood transcriptomes of blood leukocytes. Now this is the slide that tries to summarize all the gene expression data, and I depicted here again this reactome circle, where you see that the immune system is here, this triangle here, this piece of the pie, which got our most interest. And what you can appreciate from this slide then, that the Streptococcus, which is mainly related to Strep pneumo, but also other Streptococcus species, show the most profound changes in the immune system which relate to upregulated child pathways within the immune system. Now, we explained here all of the child pathways. This goes even further in a supplementary figure. But the take-home message is that when you look at immune-related genes, both relating to adaptive and innate immunity, Streptococcus species is associated with the most upregulated genes. And then CNS, so Coccula's negative staphylococci, this is not contaminant. So we selected those patients in which CNS was assigned as a causative pathogen to an infection. So these mainly related to line infections, but these were really associated with bacteremia. It's not a contaminant. And they have in the immune system, they have more down-regulated genes, you can tell. And then there's a lot of alteration genes that relate to cell cycle in different pathogens, as you can appreciate from this slide. And this has to do with cell division, mainly, and other cellular functions that I cannot discuss in detail here. So in summary, what I've showed you is that indeed, if you look at community-acquired pneumonia patients or bacteremic patients on the ICU, it turns out that the pathogen, at least in part, determines the host response. And that's I think it's relatively important information. What also stands out is that E. coli, both in the CAP cohort and in the bacteremia cohort, is associated with profound host immune activation or disturbance, you might say, and that the pathogen that is detected in bacteremic patients, for quite a large part, explains the variation in the blood transcriptomic profiles in those patients. Thank you very much for your attention.
Thanks, Tom, for the excellent presentations. Are there questions from the audience? All right. If not, maybe I can start, because there's a lot to unpick from this presentation, but you nicely zoom in to the region where we go between kind of the host characteristics and the pathogen characteristics. We try to find something in the middle, in the interaction between the two that determines the response. What in that middle ground or middle field, in the interaction between the host and the pathogen, do you think is the most interesting, the thing you'd want to investigate or you would want to eventually test out manipulating in a trial?
Yeah, the problem, as you are well aware of, so the knowledge which causative pathogen there is comes in relatively late in clinical practice. So it usually takes at least 24 hours or so before you know the pathogen. And then sometimes even you're not totally sure it is the pathogen. You only know it's gram-positive, gram-negative, and sometimes you have some more information. So whether this can be used in an immune modulatory trial, I'm not so sure of. So we did this more out of interest to see whether these bacteria in particular, so most of them are related to bacteria, are so different in their composition and also in vitro in their capacity to induce certain inflammatory responses. And the question then was in this, especially this bacteremia study, within this ICU critically ill population that is triggered by so many different insults, you might say, mechanical ventilation, pressors, whatnot, is the pathogen still important? And these samples were taken within 16 hours of admission. So there were already, in some of these patients, already interventions, and they were very sick. But still, you see these differences across pathogens. And that was actually the main message from this story.
Yeah, I think it really triggers the clinician side of me, because we all know that urosepsis with E. coli presents very differently in the ICU to pneumosepsis or meningitis with pneumococci. You know how it's going to go. You know it's different, but you just don't know what causes that.
Absolutely.
So absolutely very interesting. Other questions from the audience? I can't, yes, I see a hand here in the middle. You'll get a microphone. And in the meantime, can I maybe ask the speakers of the first session to hop onto the stage and go stand with Tom, because we do have some time to also ask questions about the other talks. So Peter and Mervyn, if you want to get up on the stage and stand with Tom, we'll have the question here in the middle first, and then maybe to the other speakers.
My question's about culture positivity. I'm at the University of Alabama at Birmingham, and I've been studying COVID and non-COVID culture positive sepsis to try and understand if it's an important predictive feature for outcomes. Do you think culture positivity, irrespective of organism type, is an important and underutilized feature for outcome prediction? Or what do you think about culture positivity in general now that you've studied it so molecularly?
Yeah, we haven't studied this in detail, and it seems that you have done so. So I cannot answer the question evidence-based from top of my head. I had the impression that culture-negative sepsis does not necessarily fare better than culture-positive sepsis, but you have found other data, and I'm not totally sure. Maybe my colleagues know?
Well, we did a study, there was a multi-center European one with the Abbott, array, which is a molecular diagnostic which picked up 1,000 bugs, and the culture-negative molecular diagnostic positive did much worse than culture-negative molecular diagnostics. So the trouble is, I think cultures are a relatively blunt tool. There are problems with molecular diagnostics, but I think overall they offer more. I don't know if you guys want to.
No, but I also think that it's really of importance, and maybe you can answer that, how you decipher that, is that the pneumococcus and the E. coli are very much related to the site of infection. And so it could not be only the pathogen, and for example, the culture negatives are more likely to be hospital-acquired infections. So there are also multiple other factors that might be the driving reason of worse outcome, for example.
With the Mars cohort, I was just curious, was there a difference in your outcomes? You went through the different transcriptional pathways that were activated, but just at culture negative versus culture positive, how did people fare?
We haven't looked at it, so that's a short answer. And Peter makes an excellent remark, obviously. So we did another story that was just published about source-specific outcomes. Same cohort, and then you see source-specific outcomes. Basically, that's true. And it probably will make a difference if an E. coli comes from the urinary tract or from the bile duct or so, or from perforated gut. That's absolutely true. And it's very difficult to correct for all of these things. So the community-acquired pneumonia story was done based on that. So to eliminate the source of infection.
These are all respiratory infections.
All respiratory, but not just blood culture positive, but with a pathogen assigned to the.
And so they were presumably mainly hospital-acquired, the E. coli ones.
No, this was all community-acquired, so that also came as a surprise to me. So if you look at intensive care populations with community-acquired pneumonia, you see many more gram-negatives than in community-acquired pneumonia that's out in the community. Or also in general wards, but you are.
That's correct.
That's what I was wondering from that presentation. Is that a marker of maybe more severe underlying disease? Patients maybe been visiting a hospital, not really admitted, so it's still community-acquired pneumonia, but visiting hospitals more often because of their chronic illness or something like that. Could I explain it, or do you have any other?
I don't know.
Nobody knows.
It's a logical explanation. Could also reflect maybe the pre-morbid condition of patients and what you say. And it could also reflect that some of these pathogens, once they induce pneumonia, it's more severe. So that's why these patients are more often on the ICU.
Yeah, so more questions and answers from this discussion, but that's usually great and a good. You had a question.
I have a question, actually. This is a more ethical question. Do you think at one stage we would reach an era based on these markers you have looked at and all these looking at outcomes to make decision earlier on, if you have, about how ceilings of care? Do you think we would reach that stage? Any?
No.
No.
The trouble is, can you be absolutely 100% sure? And that's the problem. Yes, we know when we look at a sick patient, likelihood of them living or dying, but if you put 10 in a row, can you say they've got a 1 in 10 chance, which of the 10? And that's the problem. So I think ethically, morally, we're not there yet. But dare I say, it happened in COVID though, didn't it? But if you look at the age of admission to intensive care units versus the majority of people who died, but then again, becomes a, you're fulfilling the prophecy without necessarily testing it. But that was the triage decision by and large in most countries.
I do think there will come a time that there will be like a bedside multivariable analysis of all kinds of, to make sure what phenotype is somebody and will this person react to this treatment intervention or another treatment intervention. And so that's why it's interesting to know, indeed, are the differences in the immune response spectrum based on the pathogen or based on male-female differences or etc. That will lead up, that's the whole aim, I think, that you would be able to phenotype subgroups of patients that might benefit more or less from a certain
Personalized
treatment.
medicine.
Yeah, exactly.
Absolutely. Great.
So thanks to the speakers of the first half of the session for this excellent discussion. And I think this talk about treatable traits really nicely integrates with the expertise of Manu Shankahari, who's just moved to Edinburgh about a month ago, and who'll tell us about immune system changes in sepsis and ARDS.
Lenny, thank you. Good afternoon. Hoping for my slides to come up. Oops. Great, thank you very much. The title for the talk that was given to me is Immune System Changes in Sepsis and ARDS, and I've got 12 minutes, so clearly I'm not going to do that. Part of the reason is that you've heard a lot about basic aspects of the immunology. I'm going to focus on a couple of things that we forget in basic immunology, so that there's a bit of discussion at the end. Just an acknowledgement to my fund that I'm funded through an NIHR Clinician Scientist Award still, so these are my views, not necessarily the NHS or the NIHR or the Department of Health. So when we think about sepsis and immune responses in sepsis and ARDS, the basic premise is fairly simple. You got an infection or injury, and then you get a host response that kind of goes slightly wrong or dysregulated, and then you go on to develop organ dysfunction, which is sepsis, and if it's respiratory dysfunction to a severe degree, it's ARDS, and then you move on to kind of a subpopulation of patients dying. And the basic premise of studying the immune system is that you can somehow magic up the immune system in such a way that you can improve outcome. And the reason why I'm going to combine sepsis and ARDS is because you all know the lung-safe data. If you look at the lung-safe data, nearly 60% of the etiology of ARDS is pneumonia, which is chest infection, or the remaining 16% is extrapulmonary sepsis. And the second reason is there in my first of the few slides that's going to come. So the discussion in the previous session ended with something like this. This is lovely work that we contributed to and Tom led, where we reviewed this idea that actually sepsis, ARDS, or critical illness, in my book, is not a kind of a meteor strike. There is usually a gradual deterioration in health, and we kind of loosely call this pre-sepsis trajectory. And then you have this acute insult with different pathogens, different sites of infection, a different underlying host response characteristic, causing heterogeneity that Tom kind of talked about in terms of pathogens and age, and Peter kind of highlighted that in the context of gender, sex differences. And I think if you follow sepsis patients or any critically ill patients longer term, they come back to hospital. Often 1 in 3 come back to hospital within 30 days, often with a worsening comorbidity or a new infection. That's Dr. Hallie Prescott's work, and some of my work has highlighted that problem. And then they often have residual abnormality in terms of musculoskeletal and cognitive. The ARDS work led by Margaret Herridge is a fantastic kind of summary of all of that that's happening. And this is the reason why we keep coming back to the immune system, because we believe that we can do something. So what I thought I will go through is a kind of a very basic startup of why insult from trauma or non-infectious insult and infection at the start looks very similar. And I think some of these interventions that we're going to think about has to kind of incorporate that idea. And then there are a few things about the immune system that makes me look at it differently in the context of infection versus insult. I'm just going to highlight that. I'm not going to go into deep details of that. And then into the overview of the immunology. So that's Mac to Windows formatting. So you get a pathogen. The first job of host response is to prevent the anatomical barrier, prevents any infection. And if that is a success, you've prevented your infection. That's why you got a mucosa, skin, etc. And then when this anatomical barrier is disrupted, you get infection. And then the infection then leads on to non-specific effectors of the innate immune system, primarily, things that are circulating, like complement, immunoglobulins, and other things. And often, some of these opsonizations of pathogens, your infection gets better. For example, a primary care physician giving a bit of antibiotics, they go home and they get better. And then you have a scenario where that has failed and these patients are coming into hospital. And most of our blood sampling and other things are focused on this population where the primary defense mechanisms have all gone. You're starting to get a host response, and that host response involves innate and adaptive immune system. And the insult is slightly different. Again, I think Mervyn highlighted the Glue-Grant paper on the endotoxin challenge, and they got another paper on trauma as well in JX Med. It's called Genomic Storm in Critically Ill Patients, where the idea that the injury initially gives you very similar looking immune responses to that of a burns patient or an endotoxin challenge, partly because transcriptionally all of those responses are very similar due to the innate immune system activation. So I just thought I'll answer 3 questions here, because this, I think, probably separates the infection versus injury question. So first job of the immune system is to separate identify the danger signal. And that happens through pattern recognition receptors. For example, if you look at the top left-hand corner, you have the TLR1/6 hybrid for lipoprotein, and then the TLR4 for endotoxin. And then there is a cascade of innate immune system activation that happens intracellularly, whether it be cell surface receptors or intracellular receptors. There's also a primary and secondary transcription. So here, if you look at the primary transcription is things like TNF, IL-1 beta, and then there is a secondary transcription that happens after that event, which kind of goes on to give IL-6 and other biomarkers that Tom has highlighted. So some of these early changes that you don't see in elderly patients could be related to the fact that the transcriptional responses may be relatively similar. What may be different is the inhibitory response to the transcriptional proteins, the ATF3 pathway. That may be different, and I think there is some more immunology that people can look at in that space. And the second question here is, how does it recognize self from non-self? The only reason why I put this slide up is because COVID was a good example where people started talking about autoimmunity in the near past, where people highlighted autoantibodies to certain cytokines, and probably the work from Akiko Iwasaki's group, where they talked about autoantibodies to many of the organs in the body, particularly liver, lung, and heart. And there are three things that happen here that I want to highlight. So first is, if you look at the top figure, a normal cell has got a cell surface marker that says it's self, as in don't touch me, and that essentially is almost like an inhibitory marker. When you get an infected cell or an injured cell, that changes and that progresses onto an assisted apoptosis. The figure in the middle is a senescent or an apoptotic cell, and that kind of undergoes phagocytosis. That doesn't intensify your inflammation because it's a pathway. Those two don't, because it's a pathway that's there to prevent that from happening. And the bottom bit is the necrotic cell, where there is inflammation that happens alongside injury. And the last point I want to make here is the difference between an insult and injury is right at the bottom. Where the adaptive immunity. Now, if you get an injury, and usually through an endogenous agonist like a any of the DAMs, like heat shock proteins, etc., they don't induce an adaptive immune response, as in you don't develop immunological memory, whereas from a microbial agonist, you expect to have adaptive immunity because that's part of the homeostatic response. So those are the three key points I wanted to kind of separate out between infection and insult. And then the last point about inflammation is that it's not all bad, it is essential, and the purpose slightly differs by insult, and the consequences also differ by insult. Let me give you a very simple overview. I've just listed these three things. Inflammatory trigger could be infection, injury, or as Mervyn highlighted, some of the mitochondrial tissue stress. All of them can lead on to inflammation. What starts to differ is the physiological purpose. The goal of an infection-related inflammation is to control the pathogen and kill it, and that's host defense. And then the idea that the pathological consequences that we see, lung injuries or organ dysfunction, are expected given the fact that you've got a host response. The mechanism that happens after injury is slightly different. There the goal is tissue repair. So you start to see metaplasia, fibrosis, and this literature is probably well understood in cancer, where pericancer fibrosis is a beautifully explained phenomenon due to macrophage activation. And the third is tissue stress, which is primarily adaptive. I think anybody growing up would have read Mervyn's thesis in Lancet on adaptation and hibernation. So that's an adaptive response, and there is a shift in the homeostatic set points that happen. So there is a reason why there are subtle differences, but broadly at the start they're all the same. So brief overview of the sepsis/infection immunology. This is again a figure from Tom's paper in immunity, and Thomas highlighted the idea of host-pathogen interaction, differences in types of pathogen and PAMs. I'm not going to go over it. What I do want to kind of highlight here is the differences in response, different types of response. You've got the pro-inflammatory response that involve the leukocyte activation, which I'll pick up in the next few slides, coagulation cascade and necrotic cell death, and that's a kind of the pathways that people have talked about for pyroptosis and inflammasome activation and IL-18. And then there are a bunch of anti-inflammatory pathways. Again, Thomas kind of highlighted it as part of the cytokine responses that is shown about IL-10 and other pathways. So here the cellular changes, as a very brief summary of the cellular changes. If you look at the innate immune cell, you get neutrophil activation. Neutrophil apoptosis is delayed by a few hours. There is a shift to left, which means there are immature forms of neutrophils in circulation. They degranulate quicker. So some of the challenges of PBMC-related immunology is because when you do a density separation, those immature neutrophils are within that kind of monocyte sort of area that could contribute to some of the gene expression data. And the dendritic cells and the other antigen-presenting cells have got the similar sort of problem where they've got an impaired antigen presentation. And I think Peter touched on the point about tolerance following second endotoxin challenge in his talk. And that is important when you think about subsequent insults. If you've got a critically ill patient who is immunosuppressed and has got a second gram-negative infection, they don't generate the same amount of immune response as that of the first infection that they came in with. And the lymphocytes, both B and T cells are profoundly altered, and they got apoptosis, exhaustion. There is a Th1, Th2 skewing, partly because of the cytokine responses that they produce, and there is an altered regulatory T cell population and impaired memory, and that's kind of across the board between B and T cells. So transcriptomics, again, Tom has gone into the depths of the transcriptome between pathogens. What I want to show you is the paper that Julian Knight's group and Tom's group produced a few years ago, where they took the whole blood transcriptome from Paxton tubes and did a microarray and showed essentially that you can have subpopulations of sepsis patients, and these subpopulations differ in their immune response. And this is Julian Knight's group, the paper led by Emma Davenport, where they highlight two subpopulations, with one of the subpopulations called as SRS1, which has got immunosuppression, endotoxin tolerance, and T-cell exhaustion. They seem to have a worse outcome. Their group has gone on to show that the SRS subphenotypes may respond differently to corticosteroids. And then the MAS consortium, again, produced a similar sort of analysis, but this time you got roughly 4 subpopulations of sepsis patients. And these subpopulations, again, differ between quite a number of pathways that are involved in the canonical signaling pathway. And I think I often use this slide to show one thing that I like to kind of highlight, which is this is whole blood transcriptome. Most of it is likely to be innate immune populations, particularly neutrophils, yet you see plenty of lymphocyte-related signatures. And I think ignoring the lymphocyte and the lymphopenia and exhaustion and impaired function memory is probably not a great idea. Something needs to be done about it. That's my personal opinion. Okay, and the penultimate slide before I wrap this up. So there is a relationship between organ dysfunction that you see in sepsis and ARDS and these immune responses. And I'm going to use myocardium because it is probably the easiest thing to explain. This is, again, Mervin's review with Alan Rudiger. So in sepsis, you got inflammation, and this inflammation in itself can cause intrinsic myocardial depression. Some of the early work that people have done highlights this beautifully. And the myocardial depression could be related to impaired calcium trafficking, and it could also be because of an excessive adrenergic stimulation causing adrenergic receptor level downregulation, the beta blocker hypothesis that Mervin has followed through. And there is autonomic dysfunction, and there is impaired circulation, both mechanically and functionally in terms of vasodilatation. And a couple of slides about sepsis survivors. We talk about acute illness, and we often stop there. And ARDS survivors are very similar because half of them will have infection as a starting point. And this is data from Sachin Yende's group. What they did was to do a cohort study across the US, collected blood samples in sepsis survivors over time from 0 to 3 up to 365 days. And what they did was to measure CRP, IL-6, and soluble PD-L1, which is a checkpoint molecule in circulation. And using this data, they go on to create a latent trajectory model of these patients and group the patients into a hyperinflammatory and immunosuppressed group of sepsis survivors and a kind of normalish looking sepsis survivors. And when you look at those two populations, what you see is that the hyperinflammatory immunosuppressed population seem to have a greater risk of rehospitalization and all-cause mortality at 1 year. So preliminary data, but yet, there is quite a lot of missing data in the biomarkers, as you would expect during follow-up studies. But yet again, there is a signal that some of these changes are relevant even in the survivor population. And I leave you with that concluding slide. It's rather complex, and it's worth studying. Thank you very much.
Thanks, Manu, for a great presentation. In the interest of time, I suggest that we hold any questions to the discussion at the end of the session. There'll still be the opportunity to ask Manu questions about this piece of research. I think she's gone.
Thank you. I'm not sure if Julia, our next speaker, is here. Oh, she just arrived. Julia. Julia Wendon from the UK. Immune dysfunction and liver disease. All yours.
Thank you, Chairman, ladies and gentlemen. Right, the liver is a very simple organ. I love it greatly. I'm going to get the first slide. I'm going to talk 15 minutes on no slides. Hold on. Let's go back one. Okay, so in terms of this presentation, I'm just going to walk through liver immunology, largely using a couple of reviews because I think they pull out some really salient points, and some of them link to the previous presentation about that repeated insult, perhaps determining your longer-term endpoint. This was where we were many years ago in terms of our understanding, and things have moved on a bit since then, but we still have this migratory pattern before determining where we go. In terms of liver disease, I think there is also something fascinating which very much links in terms of sepsis in this study, which is your determinant of any individual episode of sepsis is the insult, or in my case, liver failure, but also where you are in your inflammatory phenotype at the time of that insult. And that's really nicely demonstrated if you just give any of us some paracetamol. Those with a slightly higher CRP and slightly higher baseline inflammatory mediators developed a very low-level transaminitis. That was published about 5-6 years ago now. And so I think that step of where you are is really salient. This review is a few years old now, but really nicely demonstrates the intensity of the liver being at the center of all things. You can see I love the liver. But your portal vein brings inflammatory mediators from the gut. In cirrhosis and liver failure, it's often congested. Your liver itself is the home of your Kupffer cells, which are then exposed to these inflammatory mediators. Your cholangiocytes, your biliary tree, is incredibly sensitive to ischemia. It's very dependent on arterial perfusion pressure, and cholangiocyte ischemia happens early. And indeed, now in a post-COVID world, we're seeing sclerosing cholangitis, a function of that ischemia in that cohort of patients who've survived. We saw it before in severe sepsis. The liver, thus, is really involved in this inflammation. Monocytes are pulled in from your bone marrow into the liver, and they then frequently overflow through hepatic veins into your systemic circulation, causing functional immune paresis. It's complex, however, and that's all I'm saying about that. So this is a more recent review published in February of this year by various colleagues. It demonstrates that in a cirrhotic population, you have this prolonged period of minor systemic inflammation. And as your disease progresses, you start seeing more episodes of more severe systemic inflammation, largely driven by portal hypertension, so a congested gut with gut dysbiosis, which is well demonstrated in this cohort of patients. You then move into acute on chronic liver failure, where you have this pattern of very severe systemic inflammation in terms of cytokine profile, less so anti-inflammatory cytokines, but what you see is very severe impaired phagocytosis. So you get recurrent infections, more inflammation, and very much a vicious cycle. And we need to consider how we might modulate that both in the decompensated cirrhotic and in the acute on chronic liver failure patient. And this review also by one of my colleagues, Debbie Shawcross, demonstrates very elegantly the gut-brain liver interplay in terms of gut dysbiosis and mediation impacting on your microglia in terms of inflammation and depressed neurology. In very simple terms, ammonia itself limits your phagocytosis. So once your ammonia starts going above about 70, which is pretty standard for any sick cirrhotic in ITU, you start seeing impaired neutrophil phagocytosis and recurrent risk of sepsis. This old data, and this is on pure acute liver failure, although the same can be seen in acute on chronic, you see as patients develop acute liver failure, they get marked suppression of monocyte HLA-DR and increased circulating IL-10, and quite a nice relationship between IL-10 and HLA-DR suppression. Fairly simplistic in its interpretation, but it's something which is seen repeatedly, regardless of the etiology of the liver failure and the time course of the liver failure. In terms of the liver microenvironment, you see in the top left there what a necrotic liver looks like. You've got areas of necrosis, and within that necrosis, you get a pull of your monocytes from the bone marrow and innately into that necrotic area. They become anti-inflammatory, trying to resolve that inflammation, and then you run the risk of overflow. And you can see that both in terms of the hepatic cytokines, but also when you look at necrotic and non-necrotic areas, you see a differential in terms of that cytokine expression and the monocytes. And so you then end up with a model, perhaps, of an insult to the liver with sterile inflammation, central lobular necrosis. You see, in terms of that interplay between that microenvironment and your monocyte phenotype, in terms of inflammation, anti-inflammatory, and those change in monocyte functions, and demonstrable overflow into the systemic circulation in terms of that anti-inflammatory phenotype, impeding immune response and setting the patient up for repeated episodes of inflammation, failure of liver regeneration. The plasma of patients with acute liver failure is also fascinating. This is some studies done by my colleague Harry Antonides some years ago, looking at healthy monocytes incubated with acute liver failure sera. And you can see in the bottom graphs that there is an impact of acute liver failure sera in terms of both TNF suppression from those stimulated monocytes and in terms of IL-6, but actually increased IL-10 production. That led, amongst other things, to a multi-center randomized controlled trial of plasma exchange, showing resolution of many of the inflammatory abnormalities and immune abnormalities that were seen. This was 3 days of high-volume plasma exchange and an improvement in outcome in terms of non-transplanted outcome. That's now been repeated by another center and shown the same results. It's interesting that in using 100% plasma replacement, you also correct some of the coagulation defects, both the pro and the anticoagulation, and probably thus also improve some of the microparticles that are seen in acute liver failure and contribute to the tissue necrosis. This is the other study that was recently undertaken, again showing a mortality benefit for plasma exchange. It's a blunt tool, though, and I don't think more is necessarily better. We don't know when to do it, we don't know how long to do it, or what dosage to give. So coming back to cirrhosis and that disease and the review article by Rajiv Jalan and others, you can see that they demonstrate quite nicely that pathway as you go through cirrhosis, portal hypertension, activation of your renin-angiotensin system, and going through low-grade systemic inflammation into high-grade systemic inflammation and immune dysfunction and immune dysregulation. In terms of the cell markers that one sees in this regard, this is well demonstrated in this table. It's fairly detailed, and I'm not going through any great amounts, but in terms of monocytes, you see in acute on chronic liver failure, which is what we're interested in, that hits the critical care environment, a suppression of HLA-DR, increased IL-10 production, and an increase in Merck-positive subtypes. You see similarly decreased phagocytosis and increased respiratory bursts, and you see Kupffer cell production of cytokines modulating over time. And again, peripherally in your lymph nodes, you see that change in Merck populations, positive to negative, in your circulating monocytes and your fixed monocytes. So it's all set up for an escalating pattern of disease. My colleagues recently have also been looking in terms of autotaxin, LPA and LPC, and showing again that dysregulation linking to monocyte phenotype and to disease progression. And again, I think this is possibly intervenable in the future, but we're really not clear at the moment what intervening would mean in terms of outcome. What one might do in the liver might have very abnormal responses peripherally. So again, one has this pattern of disease change over time with the recurrent inflammation going to severe inflammation and the changes in cytokines, monocyte function, and neutrophil function also changing over time in this disease cohort. So what are our potential strategies in terms of managing these patients? Well, probably most importantly, trying to decrease portal hypertension with use of non-selective beta blockers, in terms of decreasing gut biosis, interventions in terms of probiotics and other things are running in trials at the moment. In terms of circulating humoral factors for acute liver failure, plasma exchange does appear to be beneficial. Whether it's useful in acute on chronic is totally up for grabs. There's studies going on at the moment. I think, again, acute on chronic liver failure is too big a cohort to be generic about. There are subgroups such as acute alcoholic hepatitis where there might be a rationale, for others less so. My liver colleagues would believe that albumin solves everything in this regard, and certainly it might have a role, but none of the studies have shown a positive outcome at the moment. In terms of looking at intervening with prostaglandins, again, some colleagues would believe that infusion of albumin is beneficial, but the multi-center trials have not changed this, and the beneficial effects potentially of beta blockers. Immune modulation, really it's still all experimental rather than at the bedside, and similarly in terms of the signaling. So, Chairman, ladies and gentlemen, I'll hold at that point.
Thank you very much. I think there's probably time for a question or two. If there's any questions from anyone from the audience?
So for acute liver failure, I think there is a rationale. The difficulty then is in timing in terms of who you transplant and who you don't. In terms of acute on chronic liver failure, I think we've got to, one, await the outcomes of the trials. Two, I think in specific subgroups who are prognosis stratified into high-risk groups of mortality, I think there possibly is a rationale, but I think we need some good randomized controlled trials before we wheel it out as something we can do, therefore we should do.
Thank you. If there's no more questions, we could move to the next speaker, please.
All right, so if there's no further questions, we'll move to the next speaker, Thierry Calandra from the University of Lausanne, who will explain everything he can explain to us in 15 minutes about the concept of trained immunity. Thierry, the floor is yours.
Well, thank you very much. Let's get the first slide. Wonderful. I have no disclosure for this lecture. So up until recently, the central dogma in immunity was that you had memory for adaptive immunity and you had no memory for innate immunity, i.e., you were to be exposed again to the same pathogen, you would go through the same process with no gain in time and no gain in response, in contrast to what we know for adaptive immunity. So it was true until recently, and I've summarized here on that slide evidence from plants, from invertebrates, from mice, and lately humans that it's perhaps not exactly like this, and there is to some extent memory as well, although perhaps differently in innate immunity. And the evidence was as follows, and I draw your attention to this wonderful article by the group of Mihai Netea, who contributed a fantastic piece of work in this area of trained immunity. In plants, we know once they are exposed, they have long-term protection against reinfection, which is named systematic acquired resistance, or SAR. And it's transmitted throughout the entire plant using either signaling molecules or pattern recognition receptors, and is associated with epigenetic changes. And it's also, which is interesting, transmitted to progeny through seeds. In Drosophila, a sublethal dose of strep pneumonia provides lifelong protection against lethal doses of strep pneumonia. Vertebrates in mice, BCG will protect the athymic nude mice against secondary lethal infection due, for example, to yeast like Candida infections. And there was an interesting observation from Carl Näslund in Sweden in 1931. Then following BCG vaccination in neonates, there was a dropout in mortality, which he considered significant in the region. And the hypothesis was that it was non-specific immunity. So graphically, if I can get the pointer, I was impressed, Tom, how you were handling the stuff. Oh yeah, good, wonderful. So as you can see here, you have the amplitude of response of the innate immune response, positive on that side, negative on that side. And up until now, we do not understand which one from the microbial products, from the microbes themselves, trigger either this trained immunity with a more robust and larger magnitude of response, in contrast to others that induce an anti-inflammatory response that will limit tissue damage. And we have to explore that further. So seminal work from Mihai Netea and colleagues here in Cell Host and Microbes about 8 years ago, upon exposure and re-exposure to Candida albicans, you can see here that these mice who have been already pre-exposed tend to be protected, where those who see Candida for the first time die. And this is associated when looked into monocytes and macrophages, pre-exposing them, waiting for a week, then re-exposing them, that there is an increase in pro-inflammatory cytokine response here, TNF, induced by various microbial products. So I will walk quickly through what Mihai and others have shown, that trained immunity is associated here with glucose pathway geared towards oxidative phosphorylation in non-trained monocyte or macrophages, and then the mice here die from a subsequent exposure to Staph aureus. Whereas if you expose this monocyte or macrophages to, for example, beta-D glucan that acts through the dectin receptor, they will change the glucose pathway towards lactate production. The trained monocytes or the trained mice are then really protected from S. aureus challenge. And this acts through dectin-1 for beta-D glucan, ACT, mTOR, and IF1 alpha. In terms of the striking other observation from Mihai's group was that this strain immunity is associated with a particular signature in terms of epigenetic remodeling or reprogramming that it's happening in these cells. And they did extensive studies to show that. Now, we spoke about monocytes and macrophages that can be trained. The same had been observed for NK cells even a bit earlier. Microglial cell can be trained as well. Hematopoietic stem cells can be trained. Skin epithelial cells and lung epithelial stem cells as well as through a number of beautiful publications. Now, work that was done at our place by Thierry Roger and his colleagues, and I'm going to summarize that in a few slides. So here is the procedure. They pre-exposed the animals, in that situation, mice, to training with beta-D glucan. A week before, re-exposed 3 days prior to the challenge. And here at the bottom part, you see what they observed. Control in red, trained mice, you see that there are differences in terms of severity scores. The control mice, higher scores. the trained mice control the bacterial inoculum. This is associated with better survival in the trained animal versus the control, and the control express more pro-inflammatory cytokine. They did the same with Staph aureus, same story again, different Kaplan-Meier curves, but again, trained animals protective compared to controls, again, controlling the burden of inflammation. And they then exposed the mice either 1 week or 9 weeks with trained immunity, and then challenged them with, again, various kind of infections. So I walk you through rapidly. This is what, oh, sorry, I should go back if I can. No, I can't. So training for either 1 week or 9 weeks with Listeria has challenged, fantastic protection here again. Pneumonia due to strep pneumonia, here again, protection. At 9 weeks, you still see the same pattern. Innate immune cells are downregulated for pro-inflammatory cytokine expression compared to the control. And the most interesting things, and this is the latest publication in collaboration with MIHI as well as Evangelos Giamarellos Bouboulis, they showed that this could be transmitted to descendant, if you will. You have here the trained mice protected compared to the control mice, and if you cross a trained male with an untrained female, you get also protection. They have not yet done the reverse, which is a bit more complicated, apparently, technically. And again, this is associated on the various panels in pink before infection, and then this light blue after infection, control versus trained. You see that there is an increase in leukocytes either before or after infection, increase in monocyte, increase in PMNs, T cells, and B cells. And they explore it even further. They really reduce the number of monocytes and macrophage using clodronate. And again, you see that you lose the protection in trained animal once they have been exposed to clodronate that reduce the number of monocytes and macrophages. And they went on to show, again, that it was dependent upon IL-1 signaling in trained immunity, because if they treat with anakinra in blue here during the training, they again lose the protection. The mortality is increasing, doesn't reach statistical significance here, but it's in a relatively low number of mice. What about humans? I mentioned this observation in Sweden in 1931 with BCG, and that's, again, the experiment that was done by Evangelos. It took 65-year-old individuals that were exposed to BCG in a double-blind fashion, and then randomized, and this was a phase 3 study. They make sure that it didn't have latent tuberculosis or immunosuppression, and they look for the number of infection over time, and they had to stop the study because they could see a benefit in total number of infection, but particularly in acute lung infection, and particularly a decrease of viral infection with a relatively low number of patients, about 70 per arm. This has been reproduced recently in a nice publication by Dr. Prentice in Lancet Infectious Disease, and this was done in Ugandan neonates who were exposed either at birth or 6 weeks thereafter to BCG, and they look at the onset of neonatal infection, and you see those who had been exposed to BCG at birth had a reduced number of infection. And later on, when the second group got the BCG at 6 weeks, there was no difference there. And again, they could show that it was associated with epigenetic reprogramming. So in conclusion, yes, there are differences between the innate and adaptive systems in terms of the specificity and the features of memory. What I showed you is that trained immunity is the capacity of innate immune cell to gain memory, and this will enhance their response upon a secondary challenge. A non-specific increase in responsiveness, broad spectrum protection against various pathogens associated with increased myelopyosis, extensive metabolic and epigenetic reprogramming, and the duration in mice is up to 9 weeks after the exposure. In humans, it has been shown now up to a year after an infection. And again, I show you the BCG example. And I would like to highlight that the work was done by Thierry Roger's team. Thank you very much for your attention.
Thank you, Thierry. Are there any questions from the audience? If no questions, let me start. Oh, sorry, Mervyn. No, go. You go first. If you speak loud, I think we can hear you.
Go, please come.
No, thank you. Thanks, Thierry.
We like a good interactive session, so this is great.
No, analogy to the heart. I don't know if you're much into cardiology, but ischemic preconditioning, and then that protects against the lethal insult, and you can do the same with, isoflurane, endotoxin, etc. So do you think organs learn to adapt to one insult, they downregulate in general? Or do you think the immune system, obviously it's different, special?
No, but as I showed on the slides, there is a broad varieties of cells that are able to be trained. So there will be probably in the various organs either blood cells or stromal cells that will be able to do exactly the same. So I think it's not a unique pattern for the human system and human cells, but the same will be shared by cells from stroma of various organs, as it is non-specific.
Thanks, and an excellent question. I think we move on to the next speaker.
And final speaker, John Marshall, to talk about how do innate immune cells respond to danger. And I have to make an apology. I have to leave at 4, so I leave it in the hand of my co-chair to close up.
Okay, well, thank you very much. So probably the most important thing that we have to do as multicellular organisms, or as the cells that comprise those organisms, is to make the difference between food and danger. I think actually the slide proportions here have cut off part of the slides. Is it possible to change them? This is a widescreen format. If not, I'll try to alert you to what's just off the right-hand side of the slide. But whether you live or die in a world where there is danger very much depends on your recognizing that danger in the first place and responding to it appropriately. So the real question here is, when you see something, whether you're a cell or a whole organism, are you looking at food? In other words, is this something you want to incorporate and grow as a result of, or is this something you have to fight against? And so at a cellular level, that is really the role of the innate immune system. It is to very rapidly make the differentiation between food and foe, and one of the critical cells that does that is the neutrophil. And so what I want to talk about in this lecture is how the neutrophil recognizes something as being potentially dangerous, and how it kind of coordinates that balance between responding aggressively and involuting and allowing homeostasis to be restored. So for a neutrophil, there's a few relatively simple things. Is whatever you see covered with antibody. If it is, you bind it to your receptors and you take it up. Is it covered with complement? Same thing. And importantly, is there evidence in the chemical makeup of that thing that you're seeing that suggests it is danger? And so the concept of DAMPs, or damage-associated molecular patterns, has arisen. Here's a really beautiful series of photomicrographs of neutrophils responding to an area of necrosis in the liver. And you can see the elegance and the precision of that process. That red mark is necrotic tissue in the liver. That's not an infection. That's host tissue that's been damaged. And the green things are neutrophils. They, over time, they recognize that there's something going on there. They move towards it, and they completely surround it and get rid of it. And then ultimately, they have to disappear somehow. They have to involute so that homeostasis can be restored. And that really is the theme of this talk. So one of the critical determinants of whether something is recognized as danger or not is, are there molecular structures, molecular patterns in that either molecule or that organism that signal danger? And how specifically, like what the general principles of signaling danger are, is not fully understood. But in aggregate, we can speak about damage-associated molecular patterns, or DAMPs. And these include things like lipids from bacteria, so a lipopolysaccharide or mannan from fungi, or lipoteichoic acid. They include some of the characteristic features of bacterial and viral RNA, but they also include a host of endogenous molecules that are normally present within the cell or that have otherwise been transformed. And so that's how the innate response to injury it actually replicates the innate response to something exogenous like a bacterial infection. So what happens here, the toll-like receptors are called toll-like receptors because they look like a receptor in the fruit fly Drosophila called toll. And there's a family of receptors that, there's at least 16 altogether in humans and mice and other species. And the basic architecture of a toll-like receptor is it has an extracellular domain, shown schematically here, that recognizes these damage-associated molecular patterns. That then links to an intracellular domain called a toll-like receptor, interleukin-1 receptor domain, which is inside the cell, and it's that domain that then starts the process of recognizing the molecule, recognizing that the external part of the receptor has been bound, and changing the conformation of the internal part, and changing gene transcription. It's a fairly fundamental picture of the way our cells respond to cytokines, to DAMPs, to many, many different things, is that the structure in the extracellular environment binds to a receptor, usually to several receptors. It induces a change in the shape of those receptors. That then recruits intracellular proteins, which in turn activate transcription factors in the cytoplasm, and that leads to new gene transcription. Importantly, the process is dependent upon changes in the secondary or quaternary structure of the receptors. So the addition of a phosphate molecule is a common one, and an enzyme that adds a phosphate molecule is called a kinase. An enzyme that takes one away is called a phosphatase. And so that these intracellular molecules, through a process of phosphorylation or dephosphorylation, interact with each other in different ways. That then removes an inhibitory influence from intracellular signaling pathways, and that then enables gene transcription to occur. This is a schematic of what happens with a toll-like receptor 4. It binds endotoxin. That brings together several tier domains, which then recruit a molecule called MAL or TIRAP, which in turn recruits other molecules such as MyD88 and SARM. MyD88 activates a pro-inflammatory response. SARM is actually an inhibitory molecule. And then if the toll-like receptor happens to be within the cell, it can activate a different pathway based on TRIF in response to viruses, and that leads to interferon expression. So a complicated and tightly regulated system. Oh, there we go. So, and what happens in a human when a toll-like receptor is activated is actually quite remarkable. This is work from the late Steve Lowry's lab by Steve Calvano, which showed when you give a human volunteer a dose of endotoxin, you actually get differential expression of over 3,700 different genes. So it really, that single process results in a repurposing of about 1/7 of the human genome. Really quite a phenomenal state of affairs. I think this... here we go. Okay, so what I want to look at then is how the neutrophil recognizes danger and how it turns itself off subsequently, because the neutrophil is a remarkably potent cell. It's the first line of defense when something is wrong within the body, but if it doesn't turn itself off, damage results, including sepsis, ARDS, and a lot of the diseases that we experience in our critically ill patients. And these are very much time dependent. So this was work by Peter Ward, who showed that if you deplete neutrophils in a mouse model of cecal ligation and puncture, if you do that before you induce cecal ligation and puncture, you have more bacteria in the circulation and you have a lower survival rate. So the neutrophil is absolutely essential to the initial response to that infection. But if you do it 12 hours after cecal ligation and puncture, you have the opposite effect. You have fewer microorganisms present, and in fact, the animals live longer. So the neutrophil is, early on, is being beneficial. Later, it's actually being detrimental. And the evolutionary structure of this whole process is really quite elegant. This is work by Paul Redmond in Ireland, who had demonstrated that when neutrophils phagocytose bacteria, they undergo programmed cell death or apoptosis. And so he got the idea to create a model where you had neutrophils invading the lung as a result of a sterile injury, ischemic reperfusion from superior mesenteric artery clamping. And what he showed is if after you did that, you instilled killed E. coli into the lung, there were fewer neutrophils in the lung. They were dying after they took up the microorganisms. Strikingly, the animals did better, so that by exposing them to a damp, it actually assisted in turning off the process that was responsible for injury, and the animals had a better survival rate. And so that gets us to this balance between responding aggressively to a threat and dying through apoptosis to limit inflammatory tissue injury. I think it's relevant in the ICU patient population because one of the characteristic features of neutrophils in critically ill patients is that they are in a basal state of activation and that they don't die. Under normal circumstances, a neutrophil is programmed to die within a matter of hours after it's released from the bone marrow. In septic patients, they survive much longer, and so they're remaining alive in an activated state. And I want to introduce you, in the last part of this talk, to a key molecule that seems to regulate that distinction between being alive and activated and dying a quiescent death. And that is the enzyme caspase 8, which is the apical enzyme involved in an extrinsic pathway of apoptosis. When it is activated by the binding of a death receptor, by a ligand, it activates intracellular caspases and the cell undergoes apoptosis. But what we have shown and others have shown is that caspase 8, its activity is subject to being phosphorylated. If you add a phosphate molecule to the tyrosine 380 in the caspase 8 molecule, it no longer is capable of being activated, and in fact, it supports cell viability. And I won't go into detail here, but not only does it not I think phosphorylation means that caspase-8 can't be cleaved. It also enables it to activate another signaling pathway in the cell dependent upon PI3 kinase. But what's interesting is, and this is work a PhD student did, is that caspase-8 seems to interact with a pattern recognition receptor, TLR4. You can see in the photomicrographs there the kind of co-localization of TLR4, which is green, and caspase-8 in red in these cells. And then in this immunoprecipitation study, you can see that caspase-8 interacts with wild-type toll receptor 4, but intriguingly not with toll-like receptor 4 that has a mutation in the TIR domain. And what's important about this mutation in the TIR domain is it's the mutation found in a mouse called the C3H-HEJ mouse, which is a mouse that is resistant to the effects of endotoxin, and it was actually the basis for the discovery of toll-like receptor 4 was study of this genetic mutation in the C3H-HEJ mouse. Amongst the other things it does is it prevents TLR4 caspase-8 interactions, and when you block those interactions, you actually block normal signaling through toll-like receptor 4. So caspase-8 plays a critical role in the viability of innate immune cells. On the one hand, it can interact with receptors of the FAS family and induce cell death. On the other hand, it can interact with toll-like receptor 4 and perhaps other toll-like receptors. It does with interleukin-1 receptor. And when it does that, it activates PI3 kinase and leads to the expression of a large number of pro-inflammatory genes, including interleukin-1 beta and the sort of secondary signal that comes from IL-1. We've developed a mouse strain where we've deleted caspase-8 in myeloid cells, so in neutrophils and monocytes. And when you do that, you can challenge these animals with cecal ligation and puncture, their survival rate is substantially improved in the absence of myeloid cell caspase-8. And moreover, the histology looks better, the gross morphology of the lungs looks better, and intriguingly, there's much lower levels of circulating bacteria following cecal ligation and puncture when you delete caspase-8. So pointing to the sort of detrimental consequences of that. So I just want to leave you with this concept, still a work in progress, but the notion that, that the differentiation between recognizing danger and restoring normal homeostasis is a very finely balanced one, and a critical protein in that process is caspase-8 through a multi-protein complex that determines whether or not the cell will undergo apoptosis or become activated and generate secondary inflammatory signals. So just to conclude, the mechanisms of activation of innate immune cells are really closely linked to the same mechanisms that cause cell death. So the two sides of the same coin are closely linked, and the caspase-8, by virtue of its ability to be phosphorylated or dephosphorylated, plays a critical role in both. And with that, I want to conclude and thank those who've done the work in this, particularly my doctoral student, Sahil Gupta. Thanks a lot.
Thanks, John. That's absolutely great and very insightful. I didn't know the term survivalome before. I know it now. Are there any questions from the audience? If there are no questions, then maybe we can kind of have all the speakers of the second session. I hope Julie is still there in the back on stage so that we can have the audience ask questions to all of them. But John, can I start with a question to you, please? So you've nicely explained the role of caspase-8, but I know you're also a fabulous trialist. We also know that we've identified deficiencies before in sepsis patients, and we've given the stuff, and it hasn't worked. So merging your hat as a trialist and a basic science researcher for caspase-8, how would you proceed investigating this?
So I think there are a couple of answers. Thanks for that question. I mean, this is all highly, highly hypothetical, and I wouldn't want to spin any more on it. One is that there are a number of different strategies for disrupting this molecular complex. It's held together by heat shock protein 90, and there are inhibitors of CHOC protein 90. It's activated by Src kinases. There are Src kinase inhibitors. It's dependent on PI3 kinase. There are PI3 kinase inhibitors. So there are ways that it can be potentially modulated. The other thing that's kind of interesting, though, from a therapeutic enrichment perspective is that amongst individuals, there's variability in the caspase 8 structure. There are different isoforms. Some people have two, a caspase 8 and a caspase 8A and B. Some people only have one. And those people who have two, and unfortunately I happen to be one of them, have a much more exaggerated inflammatory response when you challenge their cells with LPS. And it looks, we haven't got enough information yet, but we've been looking at this in COVID-19. It looks like COVID-19 may increase the expression of this two isoform abnormality and increase the likelihood. So potentially that's a relatively simple biomarker of a patient that might respond to strategies that targeted this survivalsome concept.
Thanks, John, and I'll forgive you the conflict of interest for being a research subject and an investigator at the same time.
Just my cells.
All right, are there any questions to any of the speakers? I see a question there. I think if you speak loudly, we'll hear it.
I would like to speak about selenoprotein P, which is synthesis by the liver, which go down. Selenoprotein P, perhaps you don't know it. No, it's one of the best, perhaps our best plasma antioxidant enzyme, and decrease in cirrhosis. And it's involved in liver necrosis due to dequat intoxication, paraquat intoxication. So therefore, it protects the liver and it may have a major role. But if you don't know, I skip my question.
I don't know specifically about it, so I fail at the first hurdle. Others might, I don't. But there is something fascinating that might be linked, although different, in that patients with acute liver failure and indeed cirrhotics hardly ever get ARDS. And so some of the hepatocyte growth factors appear to be protective to some of your pneumocytes. Now, that's some work that was done an awful long time ago, and it hasn't been repeated recently, to my knowledge, but it is a fantastic sort of demonstration of one organ being protected whilst others are failing.
Thanks. Any other questions from the audience? I see a question in the back. Yes, you have the microphone.
Towards Dr. Salandra's talk. Do you think there's a component of what you're seeing in the innate immunity being trained, resulting in a more regulated response to infection, i.e. not causing septic shock, and therefore your mouse aren't dying? Do you think that's a component of why the next time they see the bug, they have just a better response to it and kill it more efficiently and don't end up getting septic shock, if you understand?
Jerry, I think that's a question for you.
Yes, I'm not sure I got to the point that you wanted to raise.
So I'm wondering if the second response is just a more regulated response, i.e. it's not causing sepsis, a dysregulated response, and that's what you're seeing your innate immunity doing. It's just not going crazy the second time it sees the bug, whereas previously would have gone crazy.
Absolutely, that's the correct interpretation. They get a downregulated mode at the time of the second exposure compared to the other ones. They are able to dampen the response through this epigenetic and metabolism reprogramming that protects them to go wild and really express a lot of molecule that they would otherwise do. So your interpretation is correct.
Great, thanks.
Thanks, that's a great question. Other questions from the audience? Don't be shy. If not, then Julia, I have another question for you. I was triggered by you mentioning the research into probiotics to modify the immune dysfunction in cirrhotic patients. So the last trial on probiotics I know about in the ICU was the ProPetria trial, and I know about it because it was led from my university. And that was a trial in pancreatitis, investigating probiotics, and it showed at an interim analysis a higher mortality in the treated group, and the idea was that maybe the bacteria in the gut take up more food, and then they give ischemia of the gut. something like that. Is that something you'd be worried about in these investigations that they're planning for liver?
So I'd be absolutely worried about it, but I don't think some of my other colleagues are so worried about it. There's been some quite nice work done with rifaximin, which clearly isn't a probiotic, it's a poorly absorbed antibiotic, which does show some quite interesting data in terms of decreasing inflammatory response with a subsequent insult. But that's the only one that I'm aware of that's been published thus far. Interesting, but perhaps too fast to give everybody rifaximin quite yet.
Yes, let's wait for the data first. If there are no further questions, then thanks to the speaker for your excellent presentation and great interaction, and thanks to the audience for the listening and the questions.